Hello students, welcome back to Dr. Jaggi's Academy. Today I am going to teach about amino acids, including its classification and the structure of amino acids with respect to their acidic, basic and intermediate pH solution. So let's start. So what are the amino acids? Amino acids, as the name indicate, it contains two groups. First is amino and second one is carboxylic acid. So amino acids are the organic compounds containing both amino and carboxylic group. Amino acid, these are the building block of protein. So depending upon their position of amino group with respect to carboxylic acid, they are classified as alpha, beta, gamma amino acids. For example, so we have a 2-amino ethanoic acid, which is also known as a glycine. So in this case, the carbon which is next to the carboxylic acid is known as alpha carbon. And the amino group is attached to the alpha carbon. So that is why this acid is known as alpha amino acids. The next example is 3-amino propanoic acid. So in this case, amino group is attached to the beta carbon. So that is why this amino carboxylic compound is known as beta amino acids. The third one is 4-amino butanoic acid. So in this case, amino group is attached to the gamma carbon. So that is why this is known as gamma amino acids. So we have a different classes of amino acid depending upon the position of amino group with respect to carboxylic acid. Next one is, so what are the alpha? Generally we use the alpha amino acids. So alpha amino acids are those acids in which amino group is linked to the alpha carbon atom which is next to the carboxylic acid, and its general formula is this one. So in this case, this is the alpha carbon. On the one side of the alpha carbon atom, we have a carboxylic group. On the other side, amino group. And the third position is occupied by the hydrogen. And fourth is alkyl, that is the R group. The R group, they are different. It may be hydrogen, it may be alkyl group, it may be aryl group. So depending upon the nature of R group, we obtain different types of alpha amino acids. So alpha amino acids, if R is equal to CH3, then we get this alpha amino acids, and its name is alanine. It's also denoted by its first three letter, that is ala. If R is equal to H, then we get amino acids which is known as glycine, and it is denoted by the first three letter is glycine. So the next one is the classification of amino acids. So classification of amino acid is based on the number of amino and carboxylic group present in amino acids. So we have three types of amino acid. Number one is neutral amino acids. Second one is acidic amino acid. Third one is basic amino acid. The first of all, we will discuss the acidic amino acid. First of all, we will discuss the neutral amino acids. Neutral amino acids are those amino acids which contains amino group and carboxylic are in equal number. For example, we have two amino acids, alanine and the glycine. So in this case, these two are the neutral amino acids because they contain one amino group and one carboxylic group. So the number of amino and carboxylic both are equal. So that is why these two amino acids are neutral amino acids. The next one is acidic amino acids, in which carboxylic group is greater than as compared to the amino group. So we have another example. One is aspartic acid and second is glutamic acid. So in this case, the number of carboxylic are 1 and 2, whereas the amino group is 1. So similar in the case of glutamic acid, the number of carboxylic group is 2, 1 and 2, where amino group is 1. So in both the cases, the number of carboxylic acid are greater as compared to amino group. So that is why they are classified as acidic amino acids. The next one is basic amino acids. Are those amino acids in which the amino group is greater than as compared to carboxylic group. So we have an example, lysine and the histidine. So lysine in which the amino group is 2 and carboxylic is 1, whereas in histidine the number of amino group is 3, whereas carboxylic group is 1. So that is why they are classified as basic amino acids. The next, another classification of the amino acids, there are of two types. Number one is essential amino acid and second one is non-essential amino acids. Essential amino acids are those amino acids which can't be synthesized in our body. So that is why it must be supplied in a human diet. So whereas on the other hand, non-essential amino acids are those amino acids which can be synthesized in our body and we don't require to take in our diet. The next one is the physical properties of the amino acids. The first of all, amino acids are colorless crystalline solid. Next one, they are soluble in water. They are supremely soluble in organic solvent. Third one is high melting point, means amino acid have mostly a melting point greater than 200 degrees Celsius. All these physical properties of the amino acid, they suggest that amino acid exists as a Zwitter ion or dipolar ion. So first of all, we will discuss what is the Zwitter ion or dipolar ion. So Zwitter ion or dipolar ion means Zwitter ion is a dipolar ion which containing two poles, positive and negative. And how does it form? It is formed by the transfer of proton from carboxylic acid to the amino group. So here we have a alpha amino acid. In this case, there is a transfer of this proton from the carboxylic group to the amino group. So due to transfer of proton from carboxylic to amino, this carboxylic acid is converted into a carboxylate ion, whereas NH2 group is converted into NH3 positive due to accepting of proton H positive. So since it contains two charge, negative charge on carboxylic group and positive charge on the NH3 group. So due to presence of two polar, this is known as dipolar ion. And this dipolar ion is also known as Zwitter ion. So this is a Zwitter ion or dipole. Amino acid, they exist in the form of Zwitter ion or dipolar ion. So that is why they show the properties of, they have a high melting point, they are soluble in water, they are colorless solid. So this Zwitter ion, they act as a amphoteric in nature. Amphoteric means which act as an acid as well as a base. It means Zwitter ion act as an acid as well as base. So we have a Zwitter ion or dipolar ion of alpha amino acids. So in this case, this is NH3 positive group has a tendency to donate the H positive to form stable NH2 group. It means NH3 positive it act as a H positive donor. The substance which donate H positive act as a acid. It means Zwitter ion act as a acid due to the presence of NH3 positive, because NH3 positive has a tendency to donate the H positive. On the other hand, this is a COO negative carboxylate ion. It has a tendency to accept the H positive to form a stable COOH group. So the substance which accepts the H positive act as a base. It means Zwitter ion act as a base due to the presence of carboxylate ion. Zwitter ion act as an acid due to the presence of NH3 group. So The acidic nature of amino acid is due to the presence of NH3 positive group, and basic nature of amino acid is due to the carboxylate group in the amino acids. So next one is the effect of pH on the structure of alpha amino acids. The exact structure of the amino acid depends upon the pH of the solution in which it is dissolved. We can dissolve the amino acid in acidic solution, in basic solution, or intermediate pH solution. So first we will consider what will happen if we dissolve the amino acid in acidic solution. So if we dissolve the amino acid in acidic solution whose pH value is less than 1, then what will happen? It reacts with the H positive ion which comes from the acid. So here we have a amino acid which exists in the form of dipolar ion. So it reacts with the acid H positive. So in this case, when it reacts with the acid, then it behaves like a base. Means it has tendency to accept the H positive. So which group act as a basic character or show the basic nature? So carboxylate ion, it has tendency to accept the H positive ion to form the carboxylic acid group, COOH group. So when the dipolar ion they act with the acid, then it exists in the form of cationic due to the presence of NH3 positive, due to presence of positive charge on NH3 group. So when the amino acid dissolve in acidic solution, then amino acid they exist in cationic form. Next, when they are dissolved in a basic solution whose pH value is greater than 12, then it reacts with OH negative. So we have a dipolar ion when it reacts with the base OH negative. So when it reacts with the base, then it acts as an acid. So acidic nature of the dipolar ion is due to the presence of NH3 positive because it has tendency to donate the H positive. So here NH3 positive donate the H positive to form NH2 group. So H positive combined with OH negative to form the water. So when the dipolar ion react with the base, then it form the anionic. It exists in an anionic structure due to the presence of negative charge on the carboxylate ion. So what will happen when we dissolve the amino acid in intermediate pH value whose values lies between the 1 to 12. So in this case, when we dissolve the intermediate pH solution, then amino acid exists in dipolar ion. So in this case, it shows the equilibrium with the cationic as well as anionic form. But the concentration of anionic or cationic, they are a minimum, whereas dipolar, the concentration of dipolar ion is maximum. It means at intermediate solution, the amino acid exists in dipolar form. It exists in dipolar form mainly. The dipolar contain two pole, positive and negative. So that is why dipolar ion act as a neutral ion. So next one is what is isoelectric point. So when we dissolve the amino acid in acidic solution, then it exists in a cationic form. So after passing electric current through this cationic form, then amino acid move toward the negative electrode due to the presence of positive charge on NH3 molecule. So in this case, it moves toward the negative or cathode. In basic solution, the amino acid exists in anionic form. After passing the electric current, it moves toward the positive electrode due to anode due to the presence of negative charge on carboxylate group. So in an intermediate pH, because as we already discussed that at intermediate pH, amino acid exists in a dipolar ion. So in this case, the amino acid exists in dipolar ion which is a neutral, because their positive and negative charge are equal, so no net charge. So after passing the electric current through the dipolar ion at intermediate pH, then amino acid, they don't migrate toward any electron due to its neutral nature. They neither toward the positive electrode or neither toward the nor negative electrode. So the pH at which there is no migration of the amino acid under the influence of electric field, and this pH value is known as isoelectric point. So isoelectric point again depend upon the functional group present in an amino acid. So we have a different isoelectronic point for acidic amino acid, for basic amino acid and neutral amino acid. For neutral amino acid, the isoelectric point lies between in the range of pH value from 5.5 to 6.3. For acidic amino acid, isoelectric point lies in the range from pH 3.2 to 3.5. Whereas basic amino acid pH value lies in the 7.6 to 10.8. So these are the electric point. They are present lies for the different amino acid, neutral, acidic and basic. So in this lecture we will discuss about the amino acid and its classification, and we also we have studied about the structure of amino acid and including its isoelectric point. Thank you.